So today I'm going to talk about basically how I'm laying out my breeding program. I thought this might be relevant. I've only been here for about a year or less than a year, so I don't have too much data to share from my time at LSU, but I thought maybe some of the students in the audience would be in a similar situation in the coming years of starting a new breeding program and kind of thinking about how they want to structure that, where to begin, what kind of questions to ask. So I'm going to talk a bit about that, at least the way I've gone. So to outline my talk, give a little overview of rice in the United States, Louisiana, kind of the structure of the established breeding program there at LSU, and some of the opportunities we think that markers could play a beneficial impact in the program. And then lastly, I'll talk about some of the logistical considerations of how to integrate these new technologies into the established pipeline. First, I'll talk about the research station. I work at the H. Rouse Caffey Rice Research Station. This is part of the LSU Ag Center. This is be here like the equivalent of Geneva. It's off-site, it's off the main campus, it's about an hour and a half west of the main campus in Baton Rouge. This station, I believe, is the oldest rice station in the United States. It's founded over 100 years ago. From the beginning, it's been involved in variety development, evaluation. So there's been over 50 varieties released since the opening there. One notable achievement was the development of the Clearfield technology, herbicide rice resistance. This was later licensed to BASF, but this was discovered at the rice station. Currently, we have 50 full-time staff, including faculty and all the research associates. And the education, or the research and extension is the main focus. We do some education, have graduate students, but the main focus is extension and applied research. So you can see the general areas that we have research activities in. So I want to just put this up here. This is historical rice yields in Louisiana, and I want to show this for Louisiana. Is one, a lot of times I hear these talks, and just last week at a conference in Texas, people talk about the yield plateau and all the things we need to do. Well, we haven't experienced that yet in Louisiana. Yields have been constantly on the rise, and then this is a graph I took from a publication for all the other states as well. So you see Louisiana is this yellow line. The rate of increase has been higher here. A lot of that is attributed to the Clearfield technology. It changed how we can do rice farming because it gave us a herbicide to control one of our biggest weeds. So all the states have good increases in yield. So at least rice in the United States, we certainly are experiencing a yield plateau. This is a pedigree kind of view of all the varieties that have come out of the LSU program. The ones in, don't try to read them obviously, but the ones in green are varieties that were released, and the ones in orange are kind of founders, so new germplasm that came in. And the thing I want to point out with this is, you see, it's pretty closed. The varieties that end up making it to the market typically come from crosses that were made from other varieties that were on the market. So that's not to say we don't make a lot of crosses with other things, or in the past haven't tried to make crosses with other things. It's after you go through the years and years of testing, what ends up coming out are the crosses that were made by the elite things. So again, I just want to make that comment here, is that the program is highly focused on elite by elite crosses. And based on those two previous slides, that has been successful, and we continue to get very good gains. So there's three main classes that we breed for. The first is long grain, and that's here. That's probably 75% of our activity is in long grain rice. And then we have medium grain, which falls in, here that kind of looks like a profile of a face, I didn't realize that, but that's coincidence. These are the medium grains. So again, you see some crossing between, but for the most part, the medium grains are crossed among the medium grains. We typically will cross across the long grains and the medium grains if there's a specific trait. So for example, Clearfield originated in the long grains. When we introduce that into the medium grains, that we have to do a cross between those groups. And then the last is the aromatic. This is a specialty rice market in Louisiana and the U.S. Very small acreage. So these would be jasmine type aromatic rices. All right, so a little bit on just rice production in the United States and the market. Rice is produced in the in the south in 5 states. Up here along the Mississippi Delta, a little bit in southeast Missouri. Arkansas is the largest producer in the Delta here in Louisiana and Mississippi. And then our main region in Louisiana is where our station is southwest Louisiana. This is pretty much all rice farming there. And then along the Gulf Coast in Texas. So this is pretty much one production area, and then California has its own production area. It's kind of separate. It's hard to move germplasm for phyto reasons between the regions. So last year, acres was down a little. It was around 2.7 million acres, I believe. We produced 9 million tons of rice in the United States. 85% of the rice consumed in the U.S. was produced in the U.S. But the main market for a lot of our rice production in the United States is export. So over 50% of the rice in the U.S. is exported to over 100 countries. So it actually makes the U.S. one of the largest exporters of rice globally. Certainly not one of the largest producers, but we export a lot. Most countries don't export anything. So the export market is a very important piece for us, and something that we pay very close attention to in our breeding efforts. So the theme of this talk, as the export markets are demanding higher quality rice, as people in many countries move to cities, it's harder and harder to get them to produce, to eat rice. It's harder to prepare than a lot of other foods that you can buy packaged. So Japan and Korea, this is an issue. And as people in other countries are asking for higher quality rice, and it's a bigger and bigger demand, so more and more effort is being put on it. So now I'll just focus a little bit on Louisiana. This year we had just over 400,000 acres. That's down. Typically we're around 450 or 500,000 acres. Last year we had 83% long grain, 15% of the medium grain, and 2% of the specialty kind of aromatic rices. What I'll point out here is different than some crops is the large majority of the varieties planted in Louisiana by Louisiana producers, and actually across the South in general, come from our program or from the public program. So there's not a large industry presence in rice. There is Rice Tech, who produces hybrid seed, which is very high yielding. So they do have some of the market, but there's 2 or 3. I listed all the varieties that were over 5% of the acreage. So there's 2 or 3 Rice Tech hybrids on here, but the rest are varieties out of our research station. So what we're producing is being grown by farmers. So our focus is very much on what is going to benefit the farmers and the end users. 60% of the acreage in Louisiana is clear field, so that's that herbicide-resistant rice. It's now owned by BASF. And 25% of our acreage is hybrid. So that 25% at this time would be Rice Tech hybrids, and then 75% would be LSU varieties is what's being grown. This past year, our averages were down quite a bit, but we had an excellent ratoon crop, and I'll talk about the ratoon crop in a little bit. But basically, along the Gulf Coast, we have a long enough growing season where we can get 2 cuts. So most of the numbers reported here are just always from the first cut. I don't know why they don't add them together, but you cut it once in the end of July, and it'll grow back, and you cut it again in middle or early November before we get a frost, and you get a second crop. So this past year, our first crop was down, but often the second crop will make up for it. So when you add them together, it basically came out to be a pretty average year. So there's a little bit of flexibility in there. The yields were down because we had a really wet spring. It's hard to get into the field and fertilize and things like that. So the ratoon crop this year was 50% of what the first cut was. So it's a very nice advantage of being down there. Texas and southwest Louisiana are the only environments that we can do that in. The other thing, I just put this in here because I found it interesting, is crawfish. So the rotation crop in southwest Louisiana is crawfish. This is a big crop down here. There's actually a lot of farmers now, rice prices are not favorable, are making more of their income off of the crawfish. So it's a complete aquaculture system. So last year we had over 200,000 acres in crawfish production. The way this works is, let's say last year or this year you're growing rice. Once you put that permanent flood on in July or June, you'll seed it. So you'll dump a sack of crawfish in there. Those crawfish, while the rice is still growing, will grow. They'll start to reproduce. You'll cut your rice, and those crawfish will reproduce. You'll let them grow for a few months, and then this is now crawfish season. So January to July, they'll go in here and harvest these. So the fuel for this is the rice stubble. So when we cut the rice with the combine, we'll leave that stubble there, and that is what is the main forage to sustain the crawfish. So these are just some pictures. This would be in the middle of your rice season. You might take some crawfish from your neighboring pond. Like, again, this is a rotation, so some of your fields would be in rice and some would be in crawfish, and you would switch. So this gentleman is seeding this rice field with some crawfish, which will be harvested the subsequent year. These crawfish will grow and reproduce. They'll burrow into the ground, and then that's, I think, where a lot of the incubation or the hatching happens. And they'll come back in, and this is a field. So this would be a field that's in current production. You can see some of the rice still sticking up, but those little dots you see on there are the traps. So we'll go in. It's very manual. There's not that much mechanization in this because it's concentrated Southwest Louisiana. So they'll go through in this little crawfish boat and pull the traps and bring them. And it's primarily in the South. Louisiana, Texas is a big market for it. And I guess as you radiate out from Louisiana, it gets less and less. But you can go online and buy them and have them shipped up here overnight. And then you boil them and you eat the tails, and it's usually in a net. So anyway, I just thought it was interesting. So I'm going to shift gears a little bit here and talk about our breeding program and our targets. So I mentioned, we're really focused on supporting the growers of the state of Louisiana. And there's two main targets that people care about. It's yield and it's quality. So a lot of crops, maybe corn, primarily it's yield, and all the other things are secondary. For us, quality is just as important, and it's becoming more and more important. So these are two difficult traits to breed for. So we got to find a balance to how do we make progress in one and not shift too far. So quality is this is an example of chalk. This would be an appearance trait. This would be bad, this would be good. So grain quality is a complicated topic because it really depends on who you are speaking with. So grain quality, all these users will use that term grain quality, but depending on what circle you're in, it's a different trait that we're actually interested in. So we might say, great, we're only interested in two traits, yield and grain quality. It's not really two traits. Grain quality is many traits underneath that umbrella. So you have the farmers, millers, processors, that might be producers, Kellogg's or something like that, making Rice Krispies. So Kellogg's takes the medium grain rice for those Rice Krispies. If Kellogg's doesn't improve a medium grain variety, it pretty much kills that variety because there's not a market for it. So we need to try to consider all these variables of what is quality to people. So the traits in general, uniformity is a big one. So in a harvest, are all your seeds the same size? Are they maturing at the same time? Moisture, that's very important for a lot of these downstream things. We focus a lot on milling, which would be your total head rice. So if you have 100 grams of seed, how many are left after you mill it? And how many are broken? That's a very big one. This is one of the biggest bottlenecks in our breeding program is collecting these samples. You have to do it by hand, you have to process them, you have to weigh them. Others are grain dimensions, size and shape, appearance, so that chalk I showed on the previous slide, the color of the grain, aroma, how it cooks, how it smells, and then some of these chemistry traits, such as the amylose content, which is the texture. So if you have like a Japanese or sushi rice, it's a very low amylose, it's very sticky, and then some of the other rices were more intermediate amylose, it's less sticky. And then some of these other gel consistency, how it settles after it cooks, gel temp, how long it takes to cook. So these are all things that we need to consider. I will point out, luckily, the breeders over the last 100 years have done a very good job in basically fixing some of these traits within the elite germplasm. And I say this again to go back to why we end up always back with the same crosses is because if we crossed outside of those two crosses, all the progress that was made in these areas is now you have to select on it again. So it's very, very difficult to go outside and recover something that's going to meet all these criteria. So for example, in a long grain by long grain cross, we don't really even need to focus on size and shape. It doesn't really segregate. A lot of the amylose, it doesn't really segregate. So if you stay within the class, now you can focus most of your selection on the milling and the yield. So I just raised that point because There's a lot of great things about all this diversity that's out there, but you bring in something that has good diversity for a trait, a disease, or whatever, it often comes with bad alleles for all these other things. So if you take one step forward, you might be taking 3 or 4 steps back. So obviously, yield and quality are not the only traits that are important. So there are many other traits that impact the quality and the yield, and then there's also many traits that impact the profitability for the grower. So one huge one in southwest Louisiana is this herbicide resistance. The main weed we deal with is a wild red rice. So it's a real big challenge because all the herbicides that work on rice or on the red rice also impact our crop. So you're really limited on that. There is that clear field, but as you can imagine, we talk about weeds evolving to Roundup through mutation. You can imagine how quickly this can evolve when it just outcrosses. So the stewardship programs, you have to be very, very stern on those. But we do have fields where there is weed-resistant red rice. And actually there's a new system coming out. It's an alternate mode of action that will help those fields. But anyway, it's a complicated thing. So herbicide resistance is a almost must-have for this region. Diseases, there's probably a dozen more than I could even list that will affect us, but they're sporadic, they're not regular. We have 4 diseases that we screen with. We have a pathologist at the station, Dr. Don Groth, so he'll do all of these screenings for these 4 major diseases, insects, and then the agronomic traits, mainly standability and also that retaining capacity. All right, so here are some of the considerations, I guess, I've thought about when thinking about how I want to structure my program. And so first is the clear prioritization of the breeding targets. So what do I really need to deliver that's going to make an impact to the growers? What are the strengths and the weaknesses of the starting germplasm? So in this case, I said the breeders have done a great job on a lot of those quality traits. So, well, that's great. So I want to be careful not to disrupt the progress they've made. What are the resources required for each of these breeding activities? So I commented the milling is a big bottleneck for us. What are some of the logistical constraints? And this is the one that kind of always comes back. You have an idea, and you discuss it with some people, you think about it, and you realize logistically that's not going to work as far as in time or in labor. What are our target environments, and what are the key sources of variation in our testing program? So how many locations do I need? At what point is it overkill? If I have a finite amount of locations, which ones are the most informative? Which traits experience a lot of G by E? Those types of questions. And then finally, there's a lot of technology and shiny things out there, and then kind of always coming back and saying, okay, well, once they focus on what my end product is, worry more about getting to my goal and not so much the path I took to get there. So one way to think about these traits is basically just 2 things I think that really play a major impact in the progress you can make in a trait or the way you go about breeding for that trait. One is the genetics. So is it simple or is it complex? And the other is how well can we phenotype it? So phenotyping matter, how accurate can I measure it? How fast can I do it? What's the throughput? Those types of things. So taking these 2 things together basically gives me an idea of the relative breeding difficulty or ease. So something like herbicide tolerance, very easy to phenotype, very cheap, high throughput, single gene. It's, very efficient to breed this trait. Something like weevils, that is a complex genetics. You have to dig these things up out of the ground. It's very, very difficult to phenotype. Very difficult trait to breed for. So along those lines, we can tell you, okay, is this going to be a short-term or a long-term breeding target? And also, what is the investment for you to gain going to be? So if I look at weevil, which I said is very difficult, to make genetic gain in that is going to cost me a lot more than in herbicide tolerance. So thinking about those things. So now I'll talk about just the kind of the core product development program or variety development program that we have established currently. So basically, as we progress, the number of entries goes down as we move along. So we start with over 100,000 single, say, F2 plants. We select on a single plant basis for highly heritable traits, and then we look at by rows, and the things that get advanced from here are what go into yield tests At each stage, we're essentially looking at a plant or a row or a plot, and even on phenotype today, predicting, okay, based on how it did this year in these locations, I'm predicting it's also going to do well next year. So the phenotypes are also just predictions because they're limited. This is one location, so 1 year, one location. So there's no guarantee that how it performs this year is how it's going to perform in the future. If it was, we would test 1 year and release. So anyway, just the idea that the phenotypes are also just predictions. So as we progress with less number of entries as we move along, also the cost goes up. So the investment we make per entry goes up as we progress through this process. So when we look at, this is how our program is structured today. This is one location at the station, one location, one location. So at these early stages, we're looking at a single plant in a single year or a single row at a single location. So there's really no point in trying to say, oh, I want to select for yield or some of these complex traits, because what I see here, the odds of that translating as I move along are slim to none. So we focus on the observational type traits, so basically things that are highly heritable and things that we can look at and say, okay, this plant is too tall, this plant is too late, the grain type is off. Then as we advance, here are where we get into the traits that we're really interested in. We start using multiple locations, replicated plot data, so now we can start getting a reasonable estimate of some of these quantitative traits like yield and quality. We start getting disease scores that are over multiple years. So with this, you could say, well, maybe you could just do more here, invest more money for the phenotyping. So logistically, this is one of those things that, okay, if I had all the money in the world, or if I wanted to reallocate more resources here, we still can't test for yield and quality because it's a single row. Even if you said it was accurate, we'd have no seed left for the next year, so that we don't have enough seed at these stages to do some of these traits. So it's not just a resource or a time thing. A lot of it is a seed issue. So when we look at how we're thinking that markers can maybe play an impact on this, so again, first we're looking at this as the core breeding stages and activities we're doing, that will stay the same. We have something that works. There's successful varieties coming out. So kind of how can we layer some of these new technologies into this? So in general, at the beginning stages, we see markers can play a pretty big impact. But as we progress, it's ultimately going to be field performance. So each step, maybe the weight of how much we put towards markers versus phenotype will shift. But by the end, it's 100% field-based, and how it performs in the field is whether it's going to determine whether it's a variety or not. So one of the ways we see markers fitting into this system and playing an impact is can we start some selection at this stage on, say, the yield and the quality are two main targets. So now we're waiting to hear, but when we get here, we only have 2,000 individuals. So can we make some selections here when we have larger numbers to increase our selection intensity for those traits? The other is to increase the accuracy of some traits. So some of these early stage things, for large effect genes, can we select on a marker for a disease trait or a large effect quality trait that might be cheaper, but it actually might be more accurate. So blast is a big disease for us. Some years we don't have blast. So the F2s that year, even if you could phenotype and get a good score for cheap, if blast isn't there, there's no selection. So in that case, you'd really be increasing the accuracy from zero selection to marker-based selection. And finally is reducing the cycle time. So a lot of times you'll hear, these markers are going to reduce the time to variety a little bit, I guess, but you still need to do these years of testing in the field, and the markers are never going to replace that. So it's hard to eliminate this step, and this is a bulk of the time in the variety development process. But we can increase the cycle time from how long it takes to get something into a yield trial, and also, very importantly, how long it takes to get something where you're ready to recycle it as a parent, and that's really going to drive your rate of gain there. Okay, so what are some of the applied molecular breeding activities that we're thinking about and see fitting into this? The first is general marker-assisted selection. This is great for those large effect traits. It does require the upfront investment to identify that locus, validate that locus, validate it in your germplasm, in your environments, but But once you do all those steps, it's very efficient for these qualitative traits. One of the logistical issues with this is it requires very, very large population sizes. So if you want to end up with 100 individuals to actually test, you need to genotype a lot more to get down to that 100. So that is as an issue of how do you manage that many samples. And the other is the whole genome prediction, which most people are very familiar with here. The benefits of this is that we don't need any upfront investment. We don't need to know the specific genes. It's part of our regular breeding activity, so there's no extra phenotyping resources or things like that outside of what we routinely do. And this is very efficient for the quantitative traits. So these two things together complement each other very well as two different approaches to approach different traits with different strategies. So when we look back at this little figure with our traits, really the traits that we'll target for marker system selection would fall up here, and the prediction traits would fall down here. Now, I guess you could do predictions for all of these, but for the case of, let's say, blast versus single gene or herbicide tolerance, I don't need to genotype the entire genome to tell me that it has this one specific marker. So we'll try to fragment these things from a cost standpoint. So kind of walk through how we are initially thinking about structuring this, and we kind of go through these iterations and then go back to the logistics of does the timing work, and does it fit into this core testing process that we already have in place. So new breeding process and each of these steps. So the first thing I'll just say here is we will do a fingerprint of things after they advance from this stage to this stage. One, because the numbers are now low enough, but most importantly is because it's from here to here is where we recycle things to make new breeding crosses. So that's where we want to have a decent coverage on the fingerprint. So we walk through the marker assisted selection would really be early stage F2, maybe F2, F3 for these qualitative traits. So the numbers that we've kind of forecasted that we're going to need to really implement this is somewhere around 40,000 to 50,000 samples a year. And we can spread that out over a year, but the numbers get very big very fast. But this will allow us to not reduce any of the numbers that we put the rest of the way through the pipeline from what we're currently doing. The other is that at these stages, we already phenotype for all of our key traits. They all go into different disease nurseries, the milling, the yield, all of those traits. So we're going to phenotype these just as a normal process in the breeding program, but we'll then use this information, and that will be our training set. So again, we're not doing any extra discovery outside of our normal breeding activities, but this information will feed into a training set. The good thing about this is every year it gets updated as the relevant germplasm that's coming through. It gets updated in different allele frequencies, but also the environments. If we have a year with a lot of sheath blight versus none, it'll capture that. So we'll use that information. That will be what we use to make our predictions. And we see two ways, at least initially, of utilizing these predictions as part of this pipeline. The first is to start shifting some selection to the earlier stages in this pipeline. So like I said, normally we're selecting on, our first yield is on one location, 2 reps for milling and yield at this stage. With 2,000 entries. Can we initiate some selection at an earlier stage to make that basically that funnel larger? So in this case, for the progeny rows, we usually grow out somewhere around 50,000 progeny rows, and today we're doing that phenotypic OB selection. Is it too tall? Is it too late? Is it covered in disease? So we still will do that because there's no point in genotyping this material only to find out it's too tall. So we can see that easily. So our strategy with this is we'll still do, as we always have, the OB selections. We might look at 40,000 rows, pick those that could potentially be products for those high heritability traits, and then we're in the area of 7,500 or whatever, 96 goes into that. I think it's 7,600 or something. Rows predicted a year, so we go from 40,000 to 7,000 based on phenotype, and then how we go from this 7,500 to 2,000 would be based on the prediction. So what we're hopeful that these predictions will basically enrich the quality of yield and yeah, yield and quality that enters into that PY. We're not changing the number of entries we put into that PY. It stays the same. We're just initiating some selection a bit earlier. The other is we still can utilize some of these predictions even in the stages that we test on. So primarily, you know, especially this preliminary yield test, again, we'll have phenotype data, but it's 1 year, it's one loc. So we're going to start thinking about how do we weigh that? Do we count 60% based on phenotype, 40% based on predictions, or 80-20, or what? I think in reality, you know, we're trying to find some collaborators. We're starting some work with Susan and Jen here to look at these things. But it'll probably vary by each trait. So in a year where we have 0 disease pressure, well, it'll be 100% for that disease based on markers. In a year where we have very good disease pressure, maybe we'll put more weight behind the phenotype. So trying to figure those things out. And then the last thing I don't have on here is, maybe I do, no I don't, is population prediction. I think there's a lot of interest in exploring that down the road of can we predict which populations to make, and also how big should those populations be based on what the projected variance around a given trait for a population. All right, so the real challenge, this slide is actually pretty easy to make, but the real challenge is how do we fit all this stuff in on top of the normal activities that we're doing here. It's there's a lot of activity we're doing in the field. We have locations around the state. So how do we incorporate all this stuff without disrupting our normal everyday breeding pipeline? So we have a few different pipelines. I won't go through all the detail on this, but basically it's not feasible to send everything through one approach. We need to spread it out across the year. Also, not all populations need to get managed the same way. So for example, a lot of these crosses aren't even going to segregate for some of these large effect traits because we've already done a good job of breeding them. So there's no point in doing any kind of marker-assisted selection. So basically, we estimate, looking at our pedigrees and the material we're making a lot of crosses with, probably about half of our crosses would be suitable or reasonable to do marker-assisted selection. It's segregating for a major blast gene or a quality trait. And the other half we won't do marker-assisted selection. There's nothing of a large enough effect or a big enough economic relevance that we would make that investment. Another thing is we have a very nice winter nursery in Puerto Rico, but there's a finite number of rows. So if we go through the winter nursery, we can test just in the second year, which is very nice. And it goes, again, to turn cycles over faster. But unfortunately, we just can't logistically handle everything there. So we have some populations that would get tested, their first yield test in the third year, and then some in the second year. And that would be prioritized based on how important is that population, in how important are, what are you trying to get out, what's the gap for the growers as far as varieties. So with this, we have these things spread out, and basically this is marker-assisted selection activities here in blue. We've structured these in a way where we have a really big push for marker-assisted selection in February, where it's kind of slow for us. We start planting in March. So we have a lot of hands who can help with all that sampling. Then we have a big push in July or June. That's the busy time in the field. So really, we can't be involved in that at all. But the good thing about that is the associates who run the lab, we have unlimited student help. There's students available. Students aren't available in February, but the staff in the station can help. And then the last big push is over November, December, January, February. Again, it's a pretty slow time at the station, so there's a lot of hands on deck. So we think, we're estimating somewhere in the vicinity of 45,000 samples, 8,000 lines for whole genome genotyping. So we're up over 50,000 samples. So a big challenge with this is figuring out how are we going to structure this where we can keep all of our everyday activities going on, but yet collect that many samples, extract that much DNA, and keep everything on track. Okay, so I want to talk a little bit, so everything I've talked about now is pretty applied. We still are a very applied program, and it will remain that way. But what are some of the research questions and things that we'll have? A lot of this stuff we hope to find collaborators where we could kind of complement each other on this, and then some of the things we'll do at the station with graduate students and so forth. So the first is, again, this is our kind of core activity, our elite breeding pipeline. Well, they shouldn't have all come up at once. But anyway, thinking about germplasm, the top here is about germplasm, and thinking a lot about how do decisions I make about what crosses I make and what germplasm I bring in will affect things like how big do my populations need to be, how many markers do I need, how many traits do I actually need to select on. So we do have a great source of new elite germplasm to come in. We do a shared nursery across all the southern rice states where we all submit a few dozen of our elite lines, and everyone grows them. We share that data, and we're free to breed with those. So that is a great source for us, because if the line is elite in Arkansas or Texas, it's going to have all the same criteria for us. So we'd be much more comfortable bringing that in, but they often bring unique genetics. The other piece is this diversity in germplasm, and this is where we need to be really thoughtful about what we do. The first question, how much of a gap in my elite space is there for this trait? So, so one, if a new disease arrives, we don't have a BLB is a big disease in Asia. If that comes to the U.S. and is as devastating here, well, we're going to have to bring something in, and it might bring some bad things with it, but you just need it. But for other traits, weighing what are the pros and cons of bringing this in. The other is the architecture of the trait. If it's a single gene, it's very easy to bring it in without disrupting things. We can back cross it, markers, and we can clean up all those other traits. But when there are quantitative traits and we actually have to try to pre-breed them in or cross them in, kind of thinking about how much effort is this going to take to do this, but also what are some of the thresholds. So am I willing to sacrifice a little on grain quality to get this trait in? And that will vary again. If it's to bring BLB in, which is just, let's say, killing all the rice, you're going to be a lot more flexible to say, okay, I'll take a hit on grain quality because I need this. But if it's something else, say straight head resistance, which is a physiological disorder, say, okay, it's great, it has straight head resistance, but I'd be much more strict on how the quality of that, the grain quality before I bring it into this space. The other is around trait and marker discovery. We don't do too much discovery on our own. The public in rice is gold mine. I mean, there's so much information out there, new papers coming out all the time. So a lot of our focus is on, let's take a look at what's out there and see what would work with us. Is more around the validation, identifying the markers, making sure it works in our germplasm. Some of the questions though we ask is, what's the size of the locus, and even if I had a marker for that and I had it in my germplasm, would I allocate the resources to do the marker assisted selection for it? So asking these questions to ourselves first before we make a large time investment to do all this work, but I'm not going to actually use it in my applied marker assisted selection. We probably won't spend much time on the discovery piece. And then the last is kind of an ongoing activity, is just really trying to optimize the number of locations, how we phenotype, and just how many reps, how many measurements, where's the trade-off, at what point is it too much and you don't get any more information from those measurements. These early stages, one location, this is always going to be on the station. We can't logistically do it anywhere else. But at this stage, we're talking 3 locations. Up here, we're doing 9 locations. So of these 9 locations, which would be the best 3 for this stage? So we have really great data sets over the last 20 years to look at this stuff and try to fine-tune kind of how we allocate our resources within this space. And then there's a lot in the prediction space, and really we're looking for collaborators to help us in that area. So that's models, what's the optimal marker number, accuracy, what are the trade-offs in that space. And then finally, developing some indexes for selection. What's the best ratio of phenotype to prediction at these different stages for different traits and different years, and ultimately hoping to automate some of these activities. So a lot of this stuff kind of a 5-year goal. We hope to have a lot of this stuff answered in the next few years. So one of the enabling things that is allowing us to do these samples is the genotyping. We just purchased this whole system from LGC. It's called, they call it the SNP line. It's based on their task marker chemistry. basically it's for SNPs, small indels, and the great thing about this is it's very low cost per data point, and it's very effective when we have high samples. So like those marker-assisted selection things, talking 40-some thousand samples to run 1 or 2 or 3 markers on. So this system is very flexible for that. It's good up to a medium density marker, a few hundred markers, you can use this, and it's high throughput. So we can, in a day, do upwards of 8,000 data points. So everything I outlined today, this system has enough throughput to allow us to do this. And we have two wonderful associates who run this, so 2 people basically. And at any given time, 1 person can run the whole system. So we've been really happy with this. We just got it up about a month ago, and it's pretty much working right out of the box. One of the things we're spending a lot of time on with this is DNA extractions. So if you have a 50-cent DNA extraction and you're only going to run 1 or 2 markers on it, that really, 40-some thousand samples, that'd be $20-some thousand in DNA to run 1 or 2 markers. So we spent the last month playing with some kind of cheap and dirty DNA extractions, getting them to work with this system, and we found that they do work. We've got that to work. So our cost for a lot of these things, our sampling, we do that directly into a 96-well PCR plate, and then we extract in that plate, and that's our stock DNA plate. So the biggest bottleneck in this whole thing is collecting the leaf sample, and if anyone's got any good ideas, I haven't found anything out there. So we're just doing it with tweezers. We do it in the greenhouse. It's almost impossible to do these numbers in a rice paddy. But we can do about a plate in a half hour. The extraction is very quick. It's basically you add a buffer and incubate it for 5 minutes and add another buffer. So that is not an issue. The cost for our DNA extraction with all consumables is under 7 cents, so that's very good. And then the genotyping, we do it in 384 or 1536 plates. If you're using the 1536, it's much more cost effective. And these costs reflect everything, all the consumables, and we haven't tried to scale these costs down yet, diluting it or trying smaller volumes. We're just kind of doing it out of the box, but less than 5 cents, and that's all consumables, tips. It doesn't count time though. Okay. So our sampling, this is an issue when we started. Wow, how are we going to keep track of all these things? How are we going to collect all these things? So what we found are these trays. This tray is 1 foot by 2 foot, and it's in a 96-well format. So this tray, these are 4 96-well plates essentially. So the way we're going to track things, just basically be from tray 7, 7A, 7B, 7D, and is it an A1 or H1 or so forth. So we can track these samples pretty easily. It doesn't take up much space. As we outgrow our space, you can stack these things. They don't stay in here for too long. So the leaf goes directly into a PCR plate. This is used for the extraction. And then these 4 plates are merged into a 384 plate, and that's our working stock. So there's a perfect relation to any flat in the greenhouse that corresponds with a PCR, a 384 PCR plate in the lab. So we don't have to worry about tracking single plants or anything like that. We know from this well, okay, it came from this well here. So basically all the tracking is just, okay, we want, from the marker assisted selection, I want these 50 plants. Okay, this one is in A1 from plate A, C10 from plate B, and we don't need to track them by one. Another thing is the planting. So we just bought this off the shelf vacuum, and basically spread the seeds on there, hold it, and put it over here, and it deposits the seeds. It's not perfect. It works a lot better for the medium grains than the long grains, but it'll get you 75–80% of the way there. So you can plant one of these flats in probably 3 or 4 minutes. Because after you do this, you'll see, I don't know, not every little hole has a seed. Some have doubles, and then someone needs to go through. But when we were doing it by hand, it took 10–15 minutes. So that's what I'm saying. Every time you do something, oh, it's a great idea, we'll track these things, and then you do it. So, oh, it's going to take a whole day to do what we want to do. So luckily, there is a lot of stuff out there if you just look around, and this is a simple thing. We bought this off the shelf, but you probably could make it pretty easily as well. So this is the planting. These are the plants. We try to stay on a 7-day schedule, so we would plant. 7 days later, the plants look like this. We would sample, and then we hope to get the data back, analyzed, and the selections made within 7 days. So after 14 days, we'll go in and just pluck the plants we want to. We don't kind of keep track of them. It's just enriching out of these. We'll pluck the plants we'd like to keep. We'll transplant them into, I don't have a picture of it, but these very small tubes. And this tubes can hold 102 square feet. And again, the idea with those tubes, try to go as small as possible to give us one reliable panicle. And that's what we need. We need one panicle from this stuff because it would go out into the field into a panicle row. So logistically, so far, everything is coming together. I guess we'll see this year and the coming years. I'm sure it'll look a bit different as we move forward. But so far, I think we have a blueprint that we think is going to work, and we're going to move forward with. Okay, I'll summarize now, just about out of time. One, a lot of our breeding priorities are just determined by the market demand. So how many of our growers are affected by the trait? How much of an economic impact does that trait have? And that sets what our breeding priorities are for Louisiana. The demand for the grain quality, it's always been there, but it just continues to rise. People are demanding better rice. Last week there was a speaker at this conference we had in Texas who was saying that in the global market, rice is starting to become, just rice itself is starting to become more of a product than a commodity. So there's a really strong demand for this. We certainly need to continue or even increase our breeding focus on that, but at the same time, we need to maintain our focus on the yield. So some of these technologies, I think we're hopeful that will allow us to provide, or put more emphasis on the quality, but not sacrifice too much on the gains we're getting in yield. It's really critical, at least in our situation, to manage the genetic diversity, both maintaining what we have, so we don't want to, bottleneck what's already in that elite space, but at the same time be very thoughtful about what comes in into that elite space. These new technologies, they're all over the place. They're coming out all the time. Can't even keep up with them. Offer a lot of exciting opportunities. I think we're at this stage now where a lot of this is becoming cost effective to actually implement in a public breeding program. But we really need to be mindful that you can't slow down. You can't say, oh, well, I'll do all this, but then I'll make my crosses a year later. You can't disrupt the normal breeding cycle while integrating these things. And then finally, just a comment. The foundation of the breeding program is really going to be built on germplasm, your testing, how well you can collect data, manage your plots, and then a strong logistical infrastructure, managing your seeds, all those types of things. Really, all those things should really be well in place before going on to the next step. I guess that's just my opinion. I'm very fortunate and excited to be stepping into a program where all these things are in place. We have great staff, all the equipment, great data, managed seeds, and so this is a logical next step. But I would just say, if I was stepping into a brand new program or opening a program in a state that didn't have one, a lot of these things I talked about today, I'd probably put on the back burner, know what's on the horizon, but focus more on getting good plot data, getting the appropriate equipment, getting good staff, managing my seeds, and so forth. So with that, I'd like to acknowledge a lot of the other project leads at the faculty at the research station. Ms. Mona and Ms. Jennifer are the two associates who run the genotyping lab up for a year. These two women have run our DH lab for many years. No experience in the whole genotyping space, and they've done a wonderful job, and they're pretty much running that on their own already. And then within our breeding program, and there's one other person I should mention, Karen, but we have wonderful staff. I've learned so much from them in the last year, and will continue to rely heavily on them. But very fortunate to have a great established team already at the station. The other thing I'll mention here, we're very, very well supported by the Louisiana rice growers. Almost all of our research at the station, in one way or another, is either directly or largely supported by the Louisiana rice growers, which is also why I go back to our focus is for these Louisiana rice growers. We have a checkoff fund in Louisiana, so when you bring your rice to the mill, you pay a checkoff or a fee that goes back. Some of that money comes to us for research, and then some of that money goes to a promotion board. And rice in the U.S. has a very strong lobby. So we work directly with and for the growers, and we're very thankful to have that continued support. And having support like that really allows us to kind of plan for the future. It's there. We're very confident it will continue to be there, so we can start making investments today with the assumption that that money will continue to be there as we go forward. So with that, I'll stop and just say, if you're ever in the area or interested to come down, we have our field day. That's at the end of June, June 29th this year. It's a pretty big event. You can see all the stuff. We get usually about 400 people come out. And also, if you're interested, we release, LSU is very good. They release a lot of brochures and management tips for all sorts of things. This is our rice varieties and management tips, and you can find this if you just go on the LSU Ag Center website and type in rice varieties management tips. But we update this every year, and these are the varieties that we recommend to the growers. And there's economic breakdowns, breakpoints for inputs, costs, seed, all that. And, it's a nice thing. So with that, I'll stop and be happy to take any questions.
You discussed briefly the cooperative nurseries across states. Mm-hmm. So I assume those have been ongoing for years.
Many years, yeah.
Is there any thinking going into how they might change as a result of marker technologies? Or do you want to just stay the same, like the way they are?
Change in what sense?
Well, this is sort of a soundbite, but the idea that you replicate alleles as opposed to lines, right? And my experience with these nurseries is that everybody submits a half a dozen or whatever lines, and they get highly replicated all over the place. But what about this idea of everybody submitting many more lines? Each line would only get planted once in Texas, once in Arkansas, once in Louisiana. I don't know, something along those lines. I mean, I'm not sure if it's a good idea, but whether there's discussion.
There's no discussion of that, and a lot of that it's called Uniform Rice Regional Nursery. It's 200 entries. 80 of those entries are rep 3 times. Those are like the advanced, and then 120 are rep twice. So it comes out to 480 plots. Maybe doing something like what you're saying as an additional kind of thing, but I don't think it would replace this, because this is our way to evaluate how a variety a few years before it's released from Arkansas does in Louisiana. It would help whether or not we would recommend that. The other thing is this is our way to see experimental lines coming up through Arkansas or Texas or Mississippi or Missouri and decide, do I want to cross with that? So I think everyone would want to be able to eat the whole collection in their environment. But you're talking maybe doing something to help make an estimation set or something.
Yeah.
Yeah.
Right.
No, I think that would be I think very useful. It would just probably be something maybe we add on in addition. Yeah.
You mentioned that the importance of looking at quality characteristics to meet export demand, but you didn't mention in your slide on quality, you didn't mention nutritional quality. Do you think there's going to be an export market for, let's say, biofortified rice that contains better nutrients?
Yeah, potentially. We haven't had any feedback from the millers or the exporters whether they've had much of a demand for that. But absolutely, if that word came back from them that, hey, there's a sizable market, people are demanding this, then that certainly would go onto the list. But
I was kind of interested in, we were talking about early genotyping for just marker-assisted selections, very low coverage, wider range, and later you might do some whole genome. Are you, like, logistically, are you extracting DNA twice on the same lines, or you just extract the DNA already, so you just, and how does the price kind of balance out? Obviously it's much cheaper per individual earlier, but you are doing kind of double genotyping.
right.
We're really
Yeah.
simplistic and we're just doing whole genome all the time.
So the early stages, for a single marker, 2 loci or something, that would get extracted, I guess we could say an F2. Once those plants are transplanted, all that DNA would just get thrown away. We would need a warehouse to keep all of it. So we run that. Once those plants are transplanted, all those plates get thrown away. Those F2s would then go to be at least F3s. And let's say in that case, F3s would be the rows. We would go observe those rows, and the ones that we wanted, let's say we observed 40,000 F3 rows, and we picked 8,000 rows that we were interested in trying to predict. We would take that F3 seed, harvest it. Some of that would be stored if we ultimately decided to test it, and we might just germinate, one or two of those F4 seeds, genotype it, and use that for the predictions. So essentially, I guess you would be extracting it twice, but it would be a different generation, and we just logistically it'd be too hard to hold all those plates.
You showed a 7 cent per marker. Was that a per data point, or is that if you wanted to do 5 markers, would it be 35 cents or?
Well, so that cost of 7 cents is the extraction.
Oh.
So let's say 7 cents for an extraction.
All right.
After that, the cost per data point is let's just say 5 cents, a little less than that. So if you were going to run 1 marker on 1 individual, the total cost to you would be 12 cents. If you ran 2, you already did that cost for the DNA, it would be 17 and so.
I have a question about U.S. demand for rice, and I'm wondering what sort of rice we import into the U.S. primarily, and whether those are kinds of rice that can be grown in the Southern states.
Yeah, I think a lot, and don't quote me on this, but a lot of what we bring in are the specialty rices, the basmatis, the Thai jasmine. A lot of, I think, what people eat, kind of go back to the previous speaker, where they're from. If you have a population of people somewhere in the United States that are from one of these countries and they have their special rice or their Italian rice and all that kind of stuff. But most of the rice that's consumed in the U.S. is produced here. When the export market for some of these specialty rices, what I understand, at least what I've heard, is when LSU first came out with a jasmine, so there's a Thai jasmine, and they called it Jazzman, like jazz music. When they came out with that, it's very good rice, but on the export market, I mean, I think it grew to 40,000, 50,000 acres in Louisiana, but then the Thai rice exporters cut their price, undercut the price, and it so it knocked it back. Right now, it's a specialty thing. It's on 5,000 to 10,000 acres, and it's for specialty things, stuff you see in the grocery store where you pay a lot more for it than the big sacks you might buy. Juan?
You mentioned the opportunity in the season. Is there a genetic variation for the trait? Is that something that can be?
Yeah. There is genetic variation for it. So that is a trait, at least in South Louisiana and Texas, that could kill a variety. So there's not that many traits that will just kill a variety, but that's one. Obviously, yield and quality or others, but so it needs to have a good retooning capacity. Again, that's something that breeders have done a pretty good job on, but we've never done. There hasn't been too much work on trying to understand that. We do take on our yield trials. We do take a second. We do cut it twice.
Talk a little bit more about population prediction and why you might be using that?
Sure. I'm certainly not the best person to be talking about this, but the idea, my interest in it is we can basically simulate what a cross would look like and then predict those progeny. And I see value in that, one, to determine which crosses. I have a finite number of crosses I can make every year. And there's more crosses could be made than we can actually handle. So now we might use phenotype pedigree data to make that determination. This would just be another layer to kind of predict, even though all 4 of these lines are equally good for, let's say, all the traits. How would the combination of those alleles differ in this population? So one, it might say, well, actually, it's not that good. I won't make that cross. Or this cross is better. But another thing I'm interested in exploring is how big should my population be? Like right now, it's kind of like every population is the same size, but based on predictions, we can get an idea of, hey, this population is really good. It's got a high mean, but the variance is small. Then I might want to go forward with that population, but I don't need to plant a lot of individuals. Where the flip side, you might have another population, if the predictions say, hey, the mean is really good, it's equal to that other one, but there's a large variance around it, I might want to plant more for that one to try to get further up in that distribution. At least that's my ideas. I'm certainly not an expert.
We have a question from the webinar. Has the recent flooding damaged your regional trials or commercial rice production fields? If so, how will you deal with the loss of data?
We haven't planted yet, so last week was we planted our first trial last week, and that's a date of planting thing, so it's just we're planting early. A lot of that flooding that's happened is actually in the northwest part of the state. When I flew out yesterday, when I went to bed 2 nights ago, I thought there's no way I'm getting out of here. We're supposed to get 10 inches of rain. And when I got up in the middle of the night to the airport, had it rained. I talked to my wife yesterday. She said it never really rained. So most of that, what you're seeing on the national news, is like northwest Louisiana. We don't really grow rice up there. And even if we did, we haven't planted yet. Some people started planting last week, but it's very early.
So you mentioned that herbicide is very important, and right now we have the clear field. But to what extent are you doing exploration for novel sources of herbicide resistance to other herbicides, so that you're ready
Sure.
this is over time?
That's a very active research area. So BASF does a lot of work in that area. We have a biotech program at the station, which is essentially mutational breeding for herbicide resistance. A lot of the other universities do as well. So there's really no GMO work in rice. They had an issue with that 10 or so years ago, and the market does not want that. So BASF has a new mode of action out called Previsia. We were given that germplasm first rice to breed with that, and we hope to have a variety out next year. And that is an alternate mode of action. It's not as good as the clear field as far as the herbicide. It's not as broad spectrum. There's no residual. But if you have clear field resistant red rice in your field, this is a good option to work into that rotation. So there's another one coming out. The hope with that is, well, one, you can clear up your red rice fields, your clear field red rice. The other is it'll help doing those in rotation. So one of the weed scientists at the station, Dr. Eric Webster, he's doing a lot of work to figure out what's the best rotation to do. You do Roundup beans, clear field, Previsia. I'm just trying to figure out what's the best to sustain all of these technologies as long as possible.
One last question.
Could you maybe provide a little bit of insight into the market dynamics in the U.S. of quality between hybrid and open pollinated varieties? Because I know in some of the Asian countries where hybrid rice is trying to take off, there's issues with quality and the consumers don't like it. But in the U.S., most people are not nearly as discerning about rice. They buy rice because it's cheap. They don't really care besides that.
Yeah, that's a very good question. So the hybrids yield 20%. I mean, it's pretty much, they yield a lot more. There's a lot of issues with them. So hybrids aren't going anywhere, but at the same time, varieties aren't going anywhere for a long time. So the hybrids have that yield advantage, no doubt about it. But the quality is a huge issue. Also the height, the lodging. So there's a lot of those things that need to get straightened out in the hybrids. And I think it has actually harmed the U.S. rice export market, because we used to be like the gold standard in quality. And as more and more of this hybrid came in, we're 25% in Louisiana. Arkansas is 50%. Arkansas produces 3 times as much rice as Louisiana. So a lot of what is getting exported is hybrid, and the quality is a lot lower. And that's a few reasons. One, that uniformity I talked about. The hybrid, the hybrid itself is an F1, but the seed that goes to the mill is an F2. And a lot of these traits are controlled by the genetics of the seed. So within a lot of seed, a variety, the differences in the size and the shape and the moisture and all that kind of stuff are much, much more constant than in the hybrid. So that uniformity affects a lot of things. It also impacts the milling, because you have different sizes, so you get more breakage. Also, chalk is a big issue, that appearance thing. So the challenging thing for the hybrids is you could have 2 inbreds that look really good, but when you combine them, if there's different genetics make them look really good, and you combine them, now those things are going to segregate in the seed that goes there. So we have a hybrid program at the station run by Jim Ward, and his main focus now is to really just, the yield is there, but to try to improve those quality parameters. Rice Tech is the main company in the U.S., and they're doing a lot of great work and putting some serious effort to improve that. And they have. And the hybrids they had 10 years ago versus today are you know, orders of magnitude better in the quality. But I think what ultimately is going to happen is, again, it's a trade-off for any of it, but the people breeding hybrids are probably going to have to focus less on yield for a while and focus more on these quality traits, and then once they get those, go back to the yield. But the yield is already there. So I think as a varietal breeder, I'm not concerned. The varieties aren't going anywhere for a long time. And the hybrids, I have, the yield potential is there. So I mean, there's definitely a place for them as well. Okay, thank you.